Chika. I'm going to demonstrate how to streak. First, we have to disinfect our work surface. So this is a wire loop. We sterilize it using a flame. So we put it in the flame until it turns red hot. The tip should turn red hot. Okay, so I can put it at an angle such that it cools. Make sure that you don't put the tip down again. So I already have a pre-poured agar plate. It contains plate count agar. I'm going to divide it into 4 quadrants. Okay, those are my 4 quadrants. I'll label 1, 2, 3, and 4. Okay. So you may have a petri dish that has a mixture of microorganisms, but you may be interested in one colony. So to purify that colony, then we have to do streaking. So you get the wire loop, and then pick out the colony of interest. So in this case, I could pick out this. Make sure you work close to a flame, and then I'm going to smear it in the first quadrant. Okay, smear it in the first quadrant only, and then sterilize the wire loop again. Okay, wait for it to cool a bit. So if you want to cool it faster, you could stab it in the second quadrant. So this was the first quadrant that we have inoculated. So I could stab it in the second quadrant such that it cools. Okay. So after it has cooled, so I'm going to make streaks from the first quadrant to the second quadrant. So I just do that. You could make about 4 or 5 streaks from the first to the second, okay? And then you make sure that you sterilize the wire loop in between the streaks. Okay? So now that we have streaked the first and the second quadrant, we are going to cool the wire loop in the third quadrant that is not yet inoculated. Okay? We don't want to touch our microorganisms with a very hot wire loop and then they get burnt. So I'm going to make more streaks now from the second quadrant to the third quadrant. Okay. Sterilize the wire loop again. It's important to sterilize the wire loop in between each streak, or in between moving from one quadrant to the other, so as to be able to reduce the microbial concentration from one quadrant to the other quadrant. Okay. So we're going to cool this in the fourth quadrant. Okay. And now we're going to make streaks or lines from the third to the fourth. That should be okay. So you ensure that you sterilize the wire loop after you have done the streaking. Do not just place it back on the table or where you keep it, because it already carries microorganisms that could contaminate other surfaces or even other workers in the lab. So after having finished the streaking, we are now going to put the petri dish in the incubator at 37 degrees for 24 hours. Okay, so after incubation, we take a look at our results or the plate that we have. We notice that for the first quadrant where we first streaked, we have a very high concentration of microorganisms. But you notice that as we move to the second quadrant, which was the second quadrant, we have the colonies becoming more defined. This is more prominent. As you can see, this was the third quadrant. It's just that I removed the markings. And then in the fourth quadrant, we now have the individual isolates, or the individual isolated colonies, pure colonies, which we can now use for further studies. In cases where we have a mixture of colonies, a mixture of microorganisms, when you look at the fourth quadrant, you can actually see them well isolated. You can see one different from the other based on the morphological characteristics. It's from the fourth quadrant that we're able to pick pure isolated colonies for further analysis, such as gram staining and other biochemical analysis that we may be interested in. So from a strict plate, you may be interested in a particular colony, knowing whether it is gram positive or negative. So using a sterile wire loop, sterilize the wire loop in a flame and make sure that it's red hot. Okay. One thing to note is that the cultures that we use should be fresh cultures. They should be between 18 to 24 hours old. Older cultures usually don't bring out true characteristics when you look at them under the microscope. So I'm going to just pick. Make sure that you just get a brush of the cells. Okay? Don't get too much, that is enough. And these cells are going to be put on a slide. So we have the frosted side and the unfrosted side. So you could label here if you wish, but in this case I shall not label. So I'm going to smear the slide with the culture. Make sure that you sterilize the wire loop before you put it away. Okay, so I think we can see the culture. So we're going to heat fix it. That involves quickly passing it over the flame so that it can get stuck onto the slide. Okay, so you can do that 2 to 3 times. So we're now going to put the first stain, which is the primary stain, crystal violet, onto the culture. And we let that stain settle for 1 to 2 minutes. So the importance of this crystal violet, it acts as our primary stain. It will stain the cells, and thereafter I'll show you what the other reagents are used for. We have our water here, which we're going to use to wash off this primary stain. So after about 1 to 2 minutes, we're going to wash off this first stain using distilled water. Okay. So as you can see, the cells have been stained purple. After that, we're going to put iodine, which is the mordant. It helps us fix the primary stain. Okay. So we wait for about a minute. So the iodine and the crystal violet react to form a complex with the cells. So we wait for a few seconds, that's 20 to 60 seconds. It's very important that these reagents are added in their order. First, it's the crystal violet, followed by the iodine. After the iodine, we're going to wash this off using alcohol, which is the decolorizer. This is concentrated ethanol. We're going to decolorize the cells. Okay. So you make sure that you don't put too much alcohol, otherwise you end up with false results. Just put enough to wash off the iodine. Okay. After we have washed off after we've decolorized, then we also wash with distilled water. So we're going to end with the counter stain, which is safranin. Some people call it safranin, but it's okay. So this is a counter stain. If the cells we are staining are gram-positive, they're going to take up the color of this counter stain. Otherwise, they are going to lose that color. So we hold this for about a minute. After 1 minute, we are going to wash this off with the distilled water. Okay, so after washing with distilled water, we are going to blot dry. You can either blot dry in tissue, you can use tissue to dry it. Alternatively, you can leave it in air to dry on its own, or you can just put it very close to the flame. Okay, with time the water will dry off. After the slide has dried, it will be looked at under the microscope to determine if the cells are gram-positive or they are gram-negative. This is a special kind of microscope that is connected to a monitor. So you may decide to look through the eyepiece, or alternatively you can mount the camera and you just focus on looking at the monitor. So you mount the slide on the stage and then you try to focus the image. Okay. So when you eventually get the image in focus, you could, if the cells appear purple, then we know that they are gram-positive. If they are pink, then they are gram-negative. Okay, so after you've gotten a clear image in focus, you check and see whether the cells are positive, gram-positive, meaning they appear purple in color, as we can see here. For those that are gram-negative, they'll appear pink microscopically.